Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last class we started talking about basic statistics in R. So, we described the concept of hypothesis testing, we talked about basic statistical tests like parametric and non-parametric tests, we talked about t-tests, we talked some rank sum tests, and we have also seen how to do these tests in R. So, in this class we will continue from that and we will talk about correlation analysis and we will slowly go towards more advanced statistical tests. Okay. So, these are the basic tests that are required for doing more complex and advanced analysis. So, in this class our topic is correlation analysis and we will talk about Pearson and Spearman correlation. Okay. And again, what are the assumptions, why do you need to do these two different types of correlations, and when we should apply Pearson, when should we apply Spearman, which should be more appropriate, those kind of discussions we will go into, and then we will talk about basic XY plots in R. So, we will take some data and we will start with very simple visualizations and we will try to explain like the different components that we can customize. Okay, so, I have used some very simple visualization without explaining any parameters so far, but here we will start looking at these visualizations and we will see how we can customize different parameters of the plot. And of course, we will have some more classes where we actually look into more complex visualization techniques, and this will also be there when we start more advanced complex statistical analysis, right. So, basic plots are very important to learn before we actually go into more complex ones. Okay, so, we will start with the correlation analysis part first, and this is a very common kind of analysis that we do for many types of biological data, right. So, we have done some experiments, we have measured something and we want to see, for example, correlation between expression of two genes, right. This is something we do, or maybe we have some field data that we want to compare, right, correlation between different features from field data, and in that case also we will use some correlation analysis, right. So, what correlation analysis does is it is actually testing for a linear relationship between two variables x and y. Okay. So, positive correlation would mean, right, an increase in x will also lead to an increase in y, right. So, that is the positive correlation value, and negative correlation mean an increase in x leads to a decrease in y, right. So, we can think of this in terms of, let us say, gene expression where we have two genes x and y, and we see this expression changes as x goes up, y also goes up, and that will be a positive correlation, whereas a negative correlation would be as x goes up, y goes down, that would be negative correlation. Okay, so, as before, I think it would be helpful if we start with an example, very simple example, and again an imaginary example in this case, and we can talk about correlation, right. So, in this example we are testing, for example, whether an increase in drug dose leads to a more effective response against a disease. Okay. So, let us perhaps imagine we are testing a drug and we want to finalize the dose of that drug, right. And in this table what you have is the drug dose on the left, the first column, which tells us like what is the concentration of drug that is being used, starting from, let us say, 5 micromolar, 10 micromolar and so on. And on the second column we have efficacy, right, how effective the drug is based on their concentration, and we see these numbers again 20%, 25%, 24%, 28%, etc. Okay. So, how do we test correlation? So, there are two ways we use two types of correlation we use. One is the Pearson's correlation, right, or where we are actually correlating between absolute values, right. This is what it will look at, right, the absolute values, how they are changing between these two x and y, right, between these two columns. Whereas, we have Spearman's correlation, which is a rank correlation. Okay. So, it does not look into the correlation between absolute values, but it actually instead ranks each column, right, from 1, 2, 3, 4, etc., based on their values and sees how these ranks correlate with each other. Okay. Now, what is the difference between these two types of correlations, okay, and when we should use this, right? So, again as you probably can imagine, these are again really similar to parametric non-parametric tests. Okay. So, Pearson's correlation, actually there are some assumptions. So, the assumption is that the data come from normal distribution. Okay. So, again we have some sort of assumption about the distribution of the data. Okay. So, the data actually is being sampled from a normal distribution. The second assumption is random sampling. Okay. These data points that we are seeing, these are random samples of the population, and there is no outlier, right. So, these are the assumptions. Whereas, Spearman's correlation, because it is doing rank correlation, right, it is not looking at the absolute values, there is no such assumption about the distribution. Okay. So, there is no assumption about normal distribution or normality. The assumptions that we have are the random sampling. Okay. So, that is still there, but this is kind of a reasonable assumption, and then we have also assumption about paired observations, right. So, the x and y are paired observations. So, which is true in our case, right. So, when we are doing correlation analysis, you most of the time you will have paired observations, right. That is what you will have only. So, paired observation assumption is also valid. What you get when you do this correlation analysis, what are the results is you will get correlation coefficient, which we denote by rho or r. Okay. So, sometimes we refer to rho, right, Spearman's correlation, or r, Pearson's correlation, or sometimes we write as Rp and Rs. Okay. So, one is Spearman, another is Pearson. Whichever correlation you use, whichever correlation analysis you use, it's the value of the correlation coefficient would be between -1 and +1, right. So, higher positive value would mean there is positive correlation, and negative value would mean negative correlation, right. So, you want if you want to see like the magnitude of correlation, you have to look at the correlation coefficient. In addition, you will also get a level of significance, right. It will tell us, right, whether the correlation is significant or not, right, and based on a p value. Okay. So, again which means there is an underlying statistical test which will give us this p value. Okay. So, so let us go back to the data, right, and we will do this again, try this out in hands on, whether an increase in drug dose leads to a more effective response against a disease. Okay. So, we will try this out in the hands on, and we will also try this out in the basic XY plots, and then perhaps we will go into the real data that I showed you already, and we can use that data and we can try looking at some of these correlation values between features that we have in that data, and then we can also try some basic XY plots and see how we can customize these XY plots, how we can control different features or parameters in that plot. Okay. So, let us now go into the R, right, go into the R console, and then we will try out this correlation analysis first on this data, that on this table that I showed you, and then we will also try this on a real data set. Okay. So, let us go to the R console, and this is where we are, and what we will do is first we will define this. We are trying this correlation analysis based on the data that I defined in the presentation, right. So, let us try this out. So, these are the concentrations of the drug that we are using. Okay. Yeah. So, this is, so we have one vector that actually is the concentration, then we have the second vector which is okay. So, we have x and y, right, and we can try the correlation test, right. So, the command is correlation test core test, right, and x y. Okay. So, when you say core test x y, the default is Pearson's correlation. So, this is what you will see, right, and the correlation coefficient is given here, right. This is the correlation value is 0.995. So, there is a very high positive correlation. Of course, this is expected, right, from the data that we can see. We will take a real data and see how these correlation values look like, and then we have the p value which is highly significant, right. This is much less than 0.01 even. So, this is a very significant correlation between these x and y. What does this mean is that, yeah, so the correlation is significant, right, and the alternative hypothesis is true correlation is not equal to 0, right. So, we can accept the alternative hypothesis, we can reject the null hypothesis. The null hypothesis is that true correlation is equal to 0 and the alternative hypothesis is true correlation is not equal to 0, right. So, this is what is given here. And then we have 95% confidence interval. So, what it tells us, right, if we most of the cases this would be the range of the correlations that we will get. Okay. We can try out the Spearman's correlation now, okay, and Spearman's correlation we just have to say method equals to Spearman. So, by default is Pearson's in R, and if you want Spearman, then you have to say method equals to Spearman. So, this will give you Spearman's rank correlation. Okay. So, here again of course, rho is 1, right. This is a correlation coefficient here, rho for Spearman's correlation this is defined as rho. So, rho is 1 because there is perfect correlation and this correlation is also significant, you have p value is 0.0004, right. So, this is actually a perfect data, but this is an imaginary data. So, we will see how this holds for a real data set. What we will try is a basic xy plot with this very small number of points, and we will see how we can customize these parameters, and then we will go into the real data and we will try out those different plots. Okay. All right. So, let us try out the basic plot. So, the function that we use is simply plot x y, right, and as you can see from the plot there is almost perfect correlation between x and y. Of course, this is bare minimum, right, and some of these fonts, some of the labels, x axis labels, y axis labels, or the numbers is too small and the points also do not look very good, right. So, what you can do is, since in R we can customize all these parameters, all these factors that we have in this plot, okay, we can touch everything in here. Okay. So, let us try this out slowly. Each of these parameters will change the plot, so that you will see how this plot transform into a very aesthetically pleasing plot. Okay. So, first thing we will try is the plot function on. So, I will try this on the side, so that we can see how this is changing, okay, with the commands here. Okay. So, what we will try first is plot x y. We will use the command PCH. Okay. So, PCH will change the point type. Okay. So, for example, if we try point PCH equals to 2, you see the point type has changed into triangles. Okay. You have, let us say, PCH 4, you see only cross. Okay. Similarly, you can try different types. So, let us say PCH 12, you see this kind of, so the sign is changing, right, the points are changing. PCH 16 is actually a nice one, because it kind of gives you field circles most. So, we can use that, or PCH 20, this also gives us field circles. Okay. We want to increase the size of this. So, PCH is the controlling the point types, right, how should we represent the points. We want to increase the size of these points, right. So, the size we can increase by CEX command, let us say we make it 2. Okay. So, we increase the point size. Okay. Okay. So, this is okay. So, maybe we want to change the color as well, right. We do not want this. Okay. So, the command is color equals to blue. Okay. So, we get the blue color. Okay. So, let us try out something about the axis. Let us do something about the axis. Okay, x axis, y axis. These labels x and y, they are kind of they do not say anything, right. So, we know the x axis is the drug dose, right, and y axis is the efficacy or efficiency, right. So, maybe we can try those out, and we can say, can we label them x axis, y axis as drug dose and efficacy respectively. Okay. So, the command to do that is x label x Okay, so we see this has changed now, the x level has changed, okay, and y level is efficacy. Okay, so the y level has changed as well, but they are too small, right? So we cannot see them even. So what we can do is we can say use the same command cex dot level, and maybe we try 1.4. So above 1 means it will increase in size, below 1 means it will go down in size. Okay. So you know, now you see these are okay, much more readable. Okay, we can read the levels much more much better than before. Okay, so let's say what we want to do also is the numbers, right? The axis levels, right? The axis numbers that we have, yeah. So that those are also too small. So maybe we want to increase the size of those numbers. Okay, so we can do the same thing. Cx dot axis is equal to 1.2, right? So we have increased the size of font size of these numbers and labels, and now they are much they look much better, right? We can read them well. We want, for example, a main title as well, right? So maybe we want to say efficacy plot or something, right? Efficacy plot for drug X. So what we can do, we can say main equals to efficacy plot for drug X. Okay, right? So we have now the main title as well. Maybe we want to increase the size of that. Okay, Cx main is okay, 1.2 is not maybe enough, so 1.4, and we have increased the size. Okay, so now it looks much nicer than what we started with, and as you can see, we have a very easy way to organize, right, all these features, right, all these changes, right, in the plot. Okay, so we can actually customize every aspect of the plot. We can look at the axis levels, we can change the color of the points, you can add main title, etc. Right. So this is something very basic XY plot. Of course, we will talk about much more advanced plots as we go along. Okay, so let's now look into the real data set, the data set that we have been using for the last few classes, and we'll see how these correlation values look like, and maybe we'll try out some XY plots from there. Okay. So let's close this for now. So to close this plot, we use the command dv.off. We'll talk about how we save plots later on, again in a visualization class. How do we actually can save this as a PDF or a JPEG or a PNG, right? So that we can put it in a presentation, in a report, right, or in a manuscript. So these are very useful, right? We want to show something from our data, so we can save them and we can use them in reports, presentations, etc. Okay, so let's now look into the real data. So the data set that we have been using. So again, we use the read table command, and this is the data set. Header equals to true, and we have loaded the data. Okay, we can use this command called names data. So it will show the column names, and we can use head because there are too many columns here. So here are the some of the columns names, right? Okay, so I'll very briefly explain what these columns actually tell us, right? So because then we can interpret the results of correlation analysis as well as the plots that we will make. So the first column is gene names, right, as we have seen already, and then so the first column is gene names. So we can try this out again. Head data first column. Okay, so these are the gene names that we have, and these are alphabetically sorted. Gene name starts with A, and then we have the next column is average expression in a specific growth medium YPD. Then we have average expression in another growth medium ST. Then what we have is something called expression heterogeneity. Okay, so what this average, so this data set is derived from single cell gene expression data, right, where this has been measured in two different environments, and this DMYPD is calculating what is the level of variation, right, of expression of each of these genes across different cells. Okay. So, high DM value would mean there is a lot of variation among cells within a population, whereas low DM would mean there is lower variation. Okay. So, we have these DM values, then we have DMYPD class. It is a classification based on DMYPD. So, it is a 0 1 classification, you will see 0 1 2 etc. Then we have DMSD. So, it is again looking at expression variation in this second medium, SD medium. Okay. Now, these are the measurements that have been done experimentally, and then these average expression and DM values were calculated. Then what we have, the other features that we have in this data are actually trying to, okay, so we will look into the other features by the names command now. These different features, they are trying to predict, for example, whether any of these features actually affect the gene expression or the expression variation. Okay. So, these are again experimentally collected features, right. For example, whether some Tata box is present, whether there is some nucleosome occupancy, whether what is the nucleosome occupancy in the promoter region at certain distance upstream of the transcription start site, etc. So, then you have something called the number of regulatory TF taken from a specific database. Again, these are generated from experimental data, and then we have a lot of these transcription factor related features. We have number of transcription factor sites. Then we have this, for example, the methylation pattern, the epigenetic patterns, right, epigenetic modifications, these histone modifications, etc. All these things are there. The goal is we want to predict this average expression as well as expression variation from the other features that we have in this data set. Okay. So, to start with, what we will do is we will look into the features correlations between, for example, average expression versus some of these features, and we can also look at the correlation between, let us say, DM values, this variation or heterogeneity value, right, in expression with other features, right. So, this is something we can look at through correlation analysis, and we can also do some basic XY plots. Okay. So, let us try this out and see how this works for some of these features. Of course, we cannot do this for all features. We have more than 250 features in here. So, we will try out some features and see whether there is any correlation or not, whether the correlation is significant or not, and then we will do the plotting. Okay, so, let us do the correlation test. As I said, we can access these features, right, by these names here. So, data dollar with average expression, right. This is we can do for data frames, right. So, this is something that is possible, and then we can take any feature that we like. I do not know. So, maybe we can look into, for example, promoter. Okay, so, H3K9 this specific histone modification pattern, right, level of this histone modification, and see how this correlation test looks like. Okay, so, here are the results. This is Pearson's correlation. It says correlation is actually around 0.10. So, this is not a very high positive correlation, but the p value is significant, right. So, it is less than 0.01. So, it is quite significant, but the correlation is not that strong, right. It is 0.1 only. So, if you remember the range, it is between -1 and 1, or for positive correlation between 0 and 1. So, closer to 1 means stronger correlation, and closer to 0 means weak correlation. So, this correlation is weak, but it is significant. Now, this is something you will come across in biological data. If you have a lot of data points and you are doing this kind of correlation test or statistical test, even when there is only little correlation or the difference is very small, you might get a significant p value, right. So, that is why it is very important to also mention the effect size. Okay. So, here effect size is the correlation coefficient, right. So, correlation value is 0.1. So, the effect is actually, the correlation is actually quite weak, although significant. Okay. So, we can also try the Spearman correlation here, and the rho is 0.14. So, slightly higher than the Pearson's correlation, and you have significant p value, right. So, which means there is significant correlation, but the correlation is not very strong. So, when you are doing correlation tests, it's testing whether the correlation is 0 or not, whether it is significantly different from 0 or not. So, alternative hypothesis is that rho correlation is not equal to 0, right. So, that is the only thing it is testing. Okay. Okay, so, we now know how to do this test. We will try correlation with another parameter, for example, which I know there is some slightly stronger correlation, okay, and this is the parameter number of regulatory TFs, transcription factors. I know where we have this stronger correlation, for example, with this DM value. Okay, so, let us try this correlation and see what. Okay, so, here you see the correlation is stronger, right, even though it is not very high, it is not close to 0.8, 0.9, etc., it is still better than what we have seen earlier, right. So, it is 0.39 and so on. Okay. And again the p value is significant, it is saying p value less than this number 2.2 into 10 to the power minus 16. This is the limit, right. If p value is lower than this, then I will tell us, okay, this p value is not, p value is significant and it is actually very small. Okay. If you want to find out the exact p value, right, in this situation, if you want to find out the exact p value, how do you go about it? So, what we can do is we can store this as a vector, we can assign this test in a vector. Okay. So, in that case it will not show you any results, it will store the results in this vector called test. Okay. And then you can access these elements, you can say core test dollar p value, okay, and it will tell you the exact p value, which in this case is 5.04 into 10 to the power minus 78. Okay. So, this is very significant. Okay. And similarly, you can get the statistic, you can get the estimate. So, estimate will give you the correlation value, right, you can get the statistics. So, all these parameters are available here. So, under core test dollar, right. So, this is something you can also look at, right, and you can get all the estimates from here. Okay. So, we have seen how to do correlation tests, right, and with a real data set. What we will do now, we will plot them. Okay. So, we know there is some correlation here, although modest. Can we plot this data? Okay. So, the same command plot x y, right, and here okay. Now, we see this kind of data point, right. It seems there is correlation, but it is not clear, so not so clear here, of course. Again we can make it nicer and we can say PCH is 20, color equals blue, and this looks a bit nicer, right. We can also, one of the things you will see, like there are many points here, cluster of points that are overlapping with each other. So, if we use this kind of color, we have no way to know whether there are like how many points are there actually. So, what we can do is we can use something like this, color RGB, right, RGB scale, where we can say use the blue color. So, the first is for red, then is for green, right, RGB, then is for blue, but we can say the fourth number is a transparency, right. So, make it a bit transparent. Okay. So, 0.3 means it will be quite transparent, and you can see now, right, these points are bit transparent. So, you can kind of getting an estimate of how many points are there. For the rest of the things, you can again change in the similar way, right. We talked about this, how do you change the x level and y level, right, number of regulatory TFs, right. We can change the size, etc. We have talked about this. So, this remains the same. Only thing we have talked about, what we can do here is, because we have too many points, we can actually use the transparent points, slightly transparent points. Okay, we can make it even more transparent, and this would be more visible. Okay, so we have seen, right, how to do the statistical test, right, correlation analysis, one of the fundamental tests that we use for many applications in biological data. And then we have also talked about basic plotting functions and how to actually customize these plots, and you see how we can improve the aesthetics of these plots. Okay, so let us go back to the conclusion, right, to the presentation. And so, what we have talked about is correlation analysis, and which we are looking at from the point of linear correlation between variables. And there are two types of correlation we can report. One is Pearson's correlation, the other is Spearman's correlation, and we get correlation coefficient, which is the effect size, and then we also get a significance value, right, level of significance from p value. And then we also have looked into the hands-on of correlation test and basic XY plot, where we have seen how we can actually take very simple plots and make them very pleasing, right, to the eyes. Thank you.